Hello friends, today I want to talk something about COVID-19 test. As you all know, every living being basic cell structure, it has got nucleus, cytoplasm and cell membrane. Likewise in viruses, there is nucleus material wrapped up around the cell membrane. In COVID-19, the same nucleus material is RNA. It is called ribose nucleic acid. In eukaryotes or other cells, there is a DNA, that is deoxyribose nucleic acid. So COVID-19 SARS-CoV-2 virus has got RNA nucleic acid, positive sense RNA, around that lipoprotein material with the spikes. So what we do is actually we take out that RNA and test it. How do we do that? As you all know, in the lab we take nasopharyngeal and oropharyngeal swab. After taking nasopharyngeal and oropharyngeal swab in clean Dacron swab, we transport to the laboratory via viral transport media. In the laboratory, they remove unnecessary contaminated DNAs and other materials. Then they extract RNA. And this RNA, we don't have any devices to directly check the RNA. We need to first convert RNA into DNA. For that, we use reverse transcriptase enzyme. With this, the help of reverse transcriptase enzyme, RNA becomes complementary DNA. This complementary DNA, in the stage 2 PCR reaction, we put it in the PCR machine with the temperature setup, like a high or low temperature setup. Many copies of complementary DNAs are produced. And after the many copies of complementary DNA is produced, there is a dye probe easily detectable by the computer machine. And once we detect this, we call it as a reactive test. In this also there is a quantification. In quantitative PCR, after the detection, if your value is less than 30, means lot of viral material was there, so more infective patient, more viral load. If the value is between 30 to 40, less viral load, and these kind of cases can be even managed at home. Even if someone doesn't have any symptoms, but the CT value is less than 30, they have to be in a separate isolation facility until he recovers. And if it is more than 40, this is not an inconclusive, it is just a quantitative test. So, and there are one more tests, they are called antibody tests, where they are just like kits. You can put a small amount of blood or serum over that and they can detect antibodies, but antibodies develop only after 1 week of the infection. By the time patient might have already suffered the infection. So these are not much reliable. Some countries are using, they are mainly for the screening purposes or some other thing, but for detection even before clinical symptoms starts also, this RT-PCR comes positive. If there are any more doubts, please comment on this video.